the last session, we thought about plant nutrients and water transport in plants, focusing solely on the roots. What we want to do in this section is actually to think more about the entire plant. Now, we have to start taking up water from the roots in our most simplest pathway. So how do we get water into the roots? And then how do we get water to want to move up the plant? Remember, the most important part of this is this has to be a passive process. The plant cannot afford to expend ATP to move water. This has to be as passive as possible. So we'll look at how the plant is able to do this in a number of different ways, keeping in mind that along a few of the steps we'll have to use a little bit of ATP, but in the grand scheme of things it's not very much. So we've talked before that xylem vascular tissue is what helps us move water up the plant. They're the water conducting cells. The substance that's actually found inside the xylem is something referred to as xylem sap, and this is going to be water and some dissolved inorganic nutrients, minerals from the soil, whatever's small enough and the correct charge and polarity to be able to be moved along with the water. In most angiosperms or flowering plants, these are actually going to be made up of very small tubes within the xylem tissue that we looked at before, known as tracheids and vessel elements. If you don't remember those terms, you can look back to plant tissues. So the whole goal is to get water to move up this plant. How can we do it? Well, we have a couple of choices. The first part is actually getting water to want to move into the xylem. So we have to establish what we call root pressure. And essentially this is moving water by osmosis. If you don't remember osmosis, we'll talk about it a little bit more in a moment, but we're looking at this movement as being minimal compared to the size of most of the plants that we want to bring water to. So that's only part of the story. As you see here, I have noted the xylem sap is only going to move a couple of meters with the root pressure alone. It's going to need help. The biggest help it has is transpiration. So transpiration is literally just the evaporation of water from the leaves of the plant. Now, under normal circumstances, we would think about this as being bad, right? We're working to get all of this water up to the top of the plant, and then we're going to say it's just going to evaporate. Well, the process of evaporation helps to move this water up the plant so that what needs to be used can be used. So we're looking for mechanisms to help us do this. This evaporation is going to be one of them, this transpiration process. The root pressure will help, but we also have to rely on the basic chemical properties of water to get this all done. So water, remember, is polar. Water has an affinity or a likeness for itself. It sets up partial positive and partial negative charges in each individual water molecule that stick together. This sticking together is actually something known as cohesion. So water is cohesive. It wants to stick to itself. Well, that's great. So that means that if I can coax one water molecule into my root, others will want to follow. The other property that we have to look at is the affinity or the likeness or the attraction of water to the inside of the xylem tissue. This is actually something known as adhesion. Why two different terms? Cohesion should tell you that you have two of the same molecule, co being two. Okay? Adhesion is how we get two different kinds of molecules to be attracted to one another. When I think of this, I think of adhesive tape, like Scotch tape. I'm usually trying to stick two different things together when I use adhesive tape. So if that helps you, you can remember that way. Now, in this process, where is this all happening? Well, root pressure, of course, is happening in the roots. Okay, so we're looking for osmosis to occur. We're going to move water by osmosis into the plant, and I'll show you how we're going to do that in just a second. When we look at the actual adhesion and cohesion properties, we're inside the xylem here. Okay, so this is inside the trachid and vessel elements, moving water up the plant. All right, now the water molecules, as you see, those dotted black lines, remember, mean hydrogen bonding. That's back from biochemistry. So the dotted lines between the two water molecules is our cohesion, and the dotted lines between a water molecule and the cell wall of our xylem is adhesion. Okay, so this process of cohesion and adhesion within the xylem will be critical for us to really move a lot of water quickly. Transpiration is what is going to be the driving force for all of this. Transpiration, you can think of like drinking out of a straw. Okay, you're applying pressure at the top of the straw and bringing liquid up the straw to your mouth, right? So the plant is going to open its stomata, those holes on the undersides of the leaves, remember, surrounded in guard cells. It's going to actually allow a little bit of water to escape. As it does that, and the water evaporates, it actually sets up a pull or a suction on the water inside the xylem and helps to move it up the plant faster. So students always ask me if water really does collect in the stomata. Does the plant really do this? And if you look at the underside of a plant that's a very happy plant, in this case, a plant hanging in my kitchen window, we can see the water droplet on the end of this leaf. This is actually collected as the water has evaporated from the underside of the leaf and condensed at the tip of the leaf. So it's actually come out of the stomata and condensed at the tip of the leaf here. And this movement of water is, of course, in my plant here, not moving very far, but if you think about the sequoia that we saw in a previous session, that's moving water a huge distance, and under the similar conditions you could see water droplets on the undersides of those leaves as well. So the stomata are actually opening and closing, and the way they do that is actually through the active transport of potassium. So remember, potassium is K+, okay? In my diagram here, it's little red or pink dots surrounding our plant cell here, okay? So the guard cells are these kind of boomerang shaped things, and the stoma is the hole. Remember, it's like the pupil in your eye. It's a space, it's a hole, it's the opening itself. The guard cells are going to surround that. Now, if I want to get my guard cells to actually allow the stoma to open, I'm going to actively move potassium into the guard cells. What does this do? Well, by moving potassium in, water follows. Water is trying to dilute out the potassium in our guard cells. Okay? So if it does that, this actually causes a bowing or a bending of the guard cells that allows the stoma to open. When the guard cells get this signal that enough water has been transpired or we're under extreme drought potential conditions, the guard cells will want to close down so that the stoma will close and will minimize water loss. Well, to do that, we have to allow potassium to leave those guard cells again, and it will. It does not want to be in high concentration inside these guard cells. It will leave, and as it does, the water will follow. And you see how the boomerang or bended shape kind of shrinks down into these straight lines and actually allows the stoma to close. So in this process, we've moved a lot of things, but we're really only moving things by osmosis. So this movement, remember, by osmosis is the movement of water. And the osmosis will move in the direction of the solute, S-O-L-U-T-E, of highest concentration. So it can be salt, as in my picture here. It could be potassium, as in our guard cells. Wherever the concentration is highest of that salt, potassium solute, the water will move to try to dilute it. Now, we have to remember, in my beaker example here, it's pretty straightforward, but remember this has a semi-permeable membrane between the two beakers. The semi-permeable membrane mimics the plasma membrane of a plant cell, whether it be the guard cell of the dermal tissue of a plant, whether it be the endoderm lining the inside of our root to protect the xylem, whatever the case may be, it's still working against that semi-permeable membrane, and it's still going to try to dilute out that solute in whatever way it can. We're going to take advantage of this when we start to produce sugars, because we set up something known as the pressure flow mechanism. So this is actually looking at the cycle of water moving up the xylem on our left into our leaf cell, where we're going to do high levels of photosynthesis. High levels of photosynthesis produce high concentrations of sugar. We don't want that sugar to stay in the leaves. We need it in the rest of the plant. It has to offload into the phloem. Remember, the phloem is our sugar transporting or food transporting vascular tissue. Sugar is going to move down the phloem, and eventually what isn't used along the way in the plant will actually get stored as starch in our root cells. So as the sugar moves into the root cell, water follows. So the phloem is going to move some amount of water. This is not solid sugar pouring down the phloem, right? It's like syrup. So when we have it move into our root cells, what we're actually looking for is to keep water moving along and setting up sort of this conveyor belt or endless loop of water movement around our plant. This goes a very long way to help us transport things up and down our plant.